everyone, and welcome to today's webinar titled SARS-CoV-2 COVID-19 Emergence, Biology, and Countermeasures. This webinar is part of an ongoing coronavirus virtual webinar series. I'm Susie Valdez of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots. Now let's get started. Before we begin, I want to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you might have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. And if you have any trouble hearing this presentation, click on the Ask a Question box located on the far left of your presentation screen. As a reminder, this presentation is educational and offers free education credits. Click on the Continuing Education Credits tab located at the top right corner of the presentation window and follow the process to obtain those credits. I now present today's speaker, Dr. Mark Denison, the Director of the Division of Infectious Diseases at Craig-Weaver and the Professor of Pediatrics and Professor of Pathology, Microbiology, and Immunology, and Professor at Vanderbilt University. For a complete biography on Dr. Denison, please visit the Biography tab at the top of your screen. Dr. Denison, Mark, you may begin your presentation. Welcome, sir.
Let's see this event occurring. Certainly I have worked on coronaviruses since 1984 when I started my infectious diseases fellowship in pediatric infectious diseases. And we have been thinking about this particular event for a long time, certainly since before the SARS epidemic. Actually, myself and my colleagues had been looking at the capacity of viruses, particularly a mouse coronaviruses, to change its species specificity, at least in the lab, looking at that cell culture. So my work has been focusing on the replication, the biology, the evolution, the viral proteins responsible for replication, and then targets for antivirals. And we've been working really for the last 10 years on specific development of and preclinical testing of antivirals, including the work that has been done on remdesivir, one of the major drugs that's in trial right now. That the preclinical development was done in my laboratory and in that of my colleague, Ralph Barrick at the University of North Carolina. As I start, I want to let you know who does the work, and that's most important, and sometimes that gets left out. So I just wanted to point out the people in my laboratory as of last fall, and then some that have graduated and moved on, but with particular focus on Maria Agostini, who was the graduate student who really performed the bulk of the work that I'll describe today, along with Dr. Jim Chappell, Jennifer Gribble, and Andrea Krausers, who is the lead scientist on our antivirals program, as well as everyone else who's contributing, and all of whom are working really 7 days a week right now for the last 3 months to test these antivirals against SARS-CoV-2. We are glad that there may be an opportunity to put to use some of the things that we've been studying in such detail. And I wanted to spend the time today really maybe taking you through a little more of the details of the biology than maybe have seen on some of the other talks, and try to get a feeling for why we have talked about this virus the way we have, why our concerns are so high, and why we think it's important to really pursue a detailed understanding of the virus biology and the targets for antivirals. With a couple I'd like to give you just a couple examples of that, because of course it's not possible to be comprehensive. And certainly I'm interested in your questions and trying to see if we can address those as we go. Okay, let's see if I can change the slides. All right. So this is just mentioning our collaborators at the University of North Carolina, Chapel Hill, Ralph Barrick, Amy Sims, Tim Sheehan, and Rachel Graham, as well as at Emory, Gilead Sciences, University of Alabama, Birmingham, and then our funding, which is all from the NIH. So I don't have any direct financial conflicts. So we have been in a long-term collaboration now with Gilead and with Emory University on compounds. Okay. I just put this up because I think this isn't a surprise for anybody that's working with coronaviruses that probably made these same predictions. But really, this is just an excerpt from one of our grant applications in 2018. And I looked back to 2013 and 2008 and found the same things. And basically, the idea is that the epidemics of SARS and MERS in humans have confirmed the capacity of coronaviruses to emerge from animal reservoirs and cause human disease, no question. And that SARS-like bat viruses are currently circulating in nature that replicate in human cells, cause disease in mice, and are resistant to existing therapeutics, antibodies, and vaccines. And that has really been published as long as 2015, but before that with bat SARS-like viruses. And then finally, with increasing overlap of human and wild animal ecologies, the potential for future zoonotic coronavirus emergences is high. And this is actually the basis for why we have the research funding that we have to study these and why we've been looking at this issue for the last multiple years. This issue of the accelerating emergence of zoonotic coronaviruses. In the past, I've had to spend a lot of time talking about this because no one is familiar with it. But I put this slide up to give you a sense that really even the old coronaviruses are new. That is, that maybe the oldest ones that we know of are maybe 800 years old in humans, which from an evolutionary perspective is very, very new to the neighborhood. And then the 229, so the four now known, two of which were identified only after the SARS epidemic, NL63, HCOV229E, human coronavirus OC43, and HKU1, all of which really have, and some of which may have only been in the last 100 to 150 years. So these are really new viruses in humans. It raises the issues and questions of when does something become an endemic virus? How long does it take? When does seasonality occur? I have many strong opinions on that. But I think what's important is to look at really the viruses that are present now. SARS since 2002 and 2004, MERS 2012 to currently and ongoing, and then our novel coronavirus or SARS-CoV-2. I think a couple points here in terms of the need for antivirals, and I'll get to this in a minute. These viruses are giving us different demonstrations of the capacity of this virus family. I hesitate to use the word unique, but it might be close to a good term. With SARS-CoV-2, with SARS in 2002, 2004, as that virus emerged, people ask me the question, why did that get controlled? Why did that go away? And this one seems to be persisting and causing so much devastation worldwide. I think one of the points was that the original SARS really was focused on severe lung disease, and transmission of that virus only occurred when someone had a pneumonia or a deep-seated lower respiratory tract infection. That allowed for identification, isolation, and control. I basically say that public health was successful because the virus allowed it. MERS, on the other hand, secondly, is less transmissible, but new cases of MERS that are still arising in the Arabian Peninsula and other places still have about a 35% mortality. So a less transmissible virus, both have much higher mortality, which represents a significant concern for if that virus changed or had a significant evolutionary adaptation that allowed it to have more rapid transmission. That would be a very great concern. And then SARS-CoV-2 seems to be sort of the Swiss Army knife of emerging zoonotic coronaviruses, this capacity for spread and shed from the upper respiratory tract, from low-level infection, from pre-symptomatic infection, along with such a range of infectious illness, from mild upper respiratory to potentially asymptomatic disease to really, really severe, profound ARDS, similar to that of SARS. So it feels like these last two were sort of I hesitate to say practice runs for coronaviruses, and that this virus we're experiencing now really has the potential for really being maintained. I think just at this point, raising this issue of what will happen with this virus in terms of seasonality, I think anyone that says they know it's nonsense. Either they're not scientists or they're ignoring their own data or the data that we have, which is none. This is a new virus in humans. There are 7 billion people on the planet who are susceptible. And so the virus is evolutionarily appears to be in a very happy hunting ground in terms of humans. So this virus is not, I don't believe, is going anywhere. It has no need to become a seasonal virus at this point. There's no evolutionary selective pressure on it. That's my opinion, but that opinion is more formed on the basis of knowledge that this is a new virus, and we can't make comparisons to viruses that have been around for hundreds or thousands of years in terms of the trends and what we would expect this virus to do within the first year that it's here in humans. So where does virus come from? I'm not going to spend a lot of time on this, but I think it's important to think about this. I know you've heard it from multiple really, really good speakers and from Wong and others, but I wanted to sort of give a slightly different perspective from the perspective of the virus's capacity. I think the idea, there's two different sort of models. One is an issue of host range mutations, and that's what people tend to focus on. Is this a mutation that arose that suddenly made this virus capable? Well, I guess when you think about what a virus has to do to be successful as a zoonotic pathogen, or that means emerging into humans and becoming human disease, which is what SARS-CoV-2 is now, what does that take? Well, it has to be able to get into a host. It has to be able to overcome innate immune responses, avoid adaptive immune responses or suppress them, avoid physical barriers, replicate, transmit, or replicate to high levels, transmit, be stable in the environment, infect new hosts. All of those things suggest that a single mutation is unlikely. In contrast, what coronaviruses, the data that's emerging seems to support more the idea that in these large diverse populations of bats and other places, that you really have a vast diversity of viruses, some of which are functioning maybe as specialists, but others which are emerging and evolving as generalists. That is, that they're ready-made to move into new hosts. And what they lack is opportunity. And what we're providing for them is opportunity, both by initial opportunities for introduction into humans, and then by either the things we do by commission or omission to provide an environment that allows for the virus to really exercise its characteristics and qualities of transmission, spread, and disease. So I think that that's a more consistent with what we're seeing with the coronaviruses as they emerge. And this slide shows this. This is an old phylogeny. It's not new, and I've overlaid things that aren't meant to be accurate localizations, with the exception of a couple things. One is if you look at the things that are circled, the blue circles or shapes that contain multiple viruses, and BT always indicates a bat virus. You can see that there's different groups, and those are the beta coronaviruses in blue, the alpha coronaviruses in the green color. The ones that are circled in red, of course, are the ones that have human disease, or can have human disease, except for the ones with the bat ones up in the corner. And then our model virus, murine hepatitis virus, which is some of the virus that we use. The beta coronaviruses really seem to be where the zoonotic viruses came from. The alpha coronaviruses and some beta coronaviruses are causing devastating diseases in animals, so that's also a place where those could arise. But the point I wanted to make here is that you can see the human coronaviruses are spread out, that MERS comes from a different group than SARS, and that and the same group that SARS is in, what we call the group 2B beta coronaviruses, are also where SARS-CoV-2 is coming from. Finally, there's these other viruses up in the right corner, and I'm calling those sort of, these are the ones that we think of as those generalists that were already ready. So these were studied in the Barrick lab and other places. And these viruses, when they were resurrected, were shown to be able to infect cells in culture, human cells in culture, and to be able to infect mice, and in some cases to cause mild or more severe disease. So these are what we're sort of referring to as ready-made pre-pandemic viruses, suggesting that these generalists exist in the population and they're really looking for an opportunity, and that's all they need. I will occasionally fall prey to using anthropomorphic terminology for the virus. I try to avoid it if at all possible, because these viruses aren't really thinking about anything. The virus that's here tomorrow is the one that was successful today. So it's really more about evolutionary fitness and selection. There isn't a goal for the virus. It's not looking for an opportunity. But when there's an evolutionary niche that arises, that virus may be prepared to take advantage of it. Okay. Now I want to move on to really talk about the issue of the need for antivirals against coronaviruses. Why have we been pursuing this? And I think now it almost feels like a moot point. Of course, it's important to pursue it. But why have we been doing this for the last 10 years? Well, I think our real issue was this issue of the broad diversity of coronaviruses in bats with demonstrated ability to infect animals in culture, or infect cells in culture, these outbreak-ready viruses, that we've known this. And we've known some other things. We've known that the failure of antibodies from one virus, coronaviruses, to neutralize other coronaviruses. So human coronavirus antibodies don't neutralize SARS or MERS. Okay, animal ones from different animal species don't neutralize the human ones. Even during the SARS outbreak, there was evidence that later in the infection, that antibodies that were being made with sort of vaccine designs against the very early civet or human strains of SARS-1 were not really very good at neutralizing or stopping those strains, even that arose later in the epidemic, 6 months to 8 months later, that really had more emerged in humans and had adapted to humans. So this idea of it's important, vaccines are critical. They may be the most important thing we can do right now. But it's also possible that they may not work either now or for a very long term. So there's a possibility. So we need to have other modalities. Second, the time to develop vaccines obviously differs from the trajectory of the epidemic. Even our most optimistic estimates think about 18 months to really get something that can be made and distributed and have an impact on the epidemic. And that universal vaccines against all coronavirus groups will likely be difficult or potentially have gaps or not be possible. So certainly there's some possibility of broadly neutralizing monoclonal antibodies that could be developed against the regions of the coronavirus spike protein that might be broadly useful and on the shelf and ready to try. That's important. That should be done. And that may be one of the most important things we can do. But it's also possible that it may not work. So this idea that if you could have broadly active compounds that are targeting conserved domains in the coronaviruses, that you could have the potential for off-the-shelf use toward highly conserved functions. And that was really the goal of our research that we wanted to do. So what were our goals? Well, we're not, for those of you who are on this webinar that are looking at, that are drug development people, you will see that not a single thing that I have on this slide is really a classic drug development goal. Because I'm not a drug development person by background. I'm a basic virologist that really has been interested in the coronavirus replication. So, but I'm also an infectious disease physician. And so my goals were those of how do we think about this broadly to be prepared. So it's more about preparation than it is about specific drug development goals. But our goals have really been these issues of the idea of having broadly active against diverse coronaviruses. So the idea, we didn't want a compound that worked well against one human coronavirus or SARS alone or MERS alone. Those are important and those are interesting for development, but for the purposes of what we wanted to target for our goals, they were to look at ones that were active against diverse coronaviruses. Second, that they had a high barrier to resistance, because antiviral resistance is something that emerges rapidly. Viruses are very good at this. I'm not going to spend a lot of time today. I would like to, if we had longer, to talk about the unique superpowers of coronaviruses in terms of having the only virus family to have RNA-dependent RNA proofreading and the capacity to limit access to that. I'll mention it briefly. But so we were looking for things that had a high barrier to resistance, those that had limited genetic paths, those that had a high fitness cost. In other words, if resistance arises, that would impact the virus detrimentally. Second, that we could extend the therapeutic window for prevention or treatment. When we think about influenza and use with oseltamivir, Tamiflu, we know that you have to treat within the first 24 to 48 hours to really have an impact because of the development of late-stage end disease. Certainly, this was important in SARS models of what we've tested. The late disease is immunopathologic, right? There's a lot of interest in things like IL-6 inhibitors, steroids, other things, because this disease is an acute respiratory distress syndrome, a serious illness of the lungs that is really secondary to virus infection. It's sort of the equivalent of the body pouring gasoline on a fire to try to put it out. And so, you know, if I think of an analogy of if I fell in a fire and burned my arm, someone came along with a fire extinguisher, they could put out the fire, but they aren't going to heal the burn on my arm. And I think a little bit about this virus that way. So if there's things that we can do to lessen the fire, if there's things that we can do to limit the damage that's being caused, along with therapeutic interventions, we can make a difference. And then finally, are there things we can do to have compounds that actually work in multiple ways? So they're not only treating infection or lessening the severity of disease, but they also may be decreasing transmission. So is there something we can do that will damage the virus in such a way that the virus that is released is less transmissible or less infectious? And those theoretically in limited environment could have an effect on local transmission of virus within a family, within a nursing home, within another kind of environment where there might be a high level of exposure. Okay, so I'm going to talk today really what I call direct-acting antivirals. Those are things that are really, you know, for treatment, prophylaxis, or transmission prevention that act directly on virus replication in some way, prevention of virus infection. Monoclonal antibodies and host-directed therapies are profoundly important, as are vaccines, but that's not really my goal to talk about those today. You can get that information from other places, and there's other great investigators. But I want to take you now briefly through the coronavirus life cycle and talk about a few of these stages and spend a little time on it. The coronavirus replication cycle is like, in some ways, it's like every other RNA virus. That is, the virus binds to receptors, in this case, angiotensin-converting enzyme 2, or ACE2. The virus enters either by direct fusion or by endocytosis. We don't know which this virus does yet. Many coronaviruses can do both, and under selective pressure, they can change their mode. So that has some impact on how we might think about blocking virus entry. Once the virus enters the cell, then the next thing that happens is the viral genome is released. It's RNA. It looks like an mRNA. It's translated into proteins. And those proteins are then processed into individual proteins from one very large replicase polyprotein of 16 protein domains that are processed by viral proteases into individual functions that function together to form virus factories on host membranes. Those host membranes, those factories then replicate the viral RNA genome, and then they replicate subgenomic RNAs that are used for making structural proteins, and those all assemble in the ERGIC or Golgi, and then they are packaged and released from the host cell. Most coronaviruses can release a lot of virus without ever lysing or damaging a cell. So that's why they're capable of really producing a tremendous amount of virus burden, because they don't really immediately kill the cells, and so that they can generate a tremendous amount of virus particles. All right. So I'm not going to spend a lot of time here because this really is getting it, but there are targets. Of course, the spike glycoprotein, which gives the coronavirus its name on the crown of spikes on its surface, is a key target is the one responsible for host receptor binding and entry, determines the species specificity, tropism, and immunity. And also the area where you have issues with lack of cross protection, because you have variability between the spike, its receptors, and its proteins on the surface, its epitopes, the regions that bind to the host receptor. I'm noticing here on the schematic on the right, the blue and green one of the spike, you can see that there's a little thing called FP. That's the fusion protein. And this is showing the fusion protein in sort of its pre-fusion form, which is the form of the protein that is being expressed in the Moderna RNA vaccine that's currently in trial. So this would be, ideally, this form of the spike protein would be a key target for vaccines to give a really good immune response that might be protective and might not cause any aberrant problems. Okay, so the other place that people are interested, of course, is ACE inhibitors. That is, those things that might bind to to ACE2, to angiotensin converting enzyme 2, and compete with the virus. I think there are many of these that are available that are used for multiple functions, and many of which could be repurposed. The challenge is that they might not bind the same place on ACE2 as the virus does. So trials, preliminary trials in our hands of a few of these have been unsuccessful. Trials elsewhere, SARS, were not very successful, and I think they're worth trying, but I think that the data to date suggests that they're not likely to be highly efficacious. But that remains to be tested, both in the lab and against the specific virus, and potentially in people, if there's good evidence that that's worth moving forward. Chloroquine, of course, is getting a lot of press. I won't spend any time on it. I have opinions, but I'm not going to share them widely here. There's not a lot of data yet that is directly antiviral. Where it might be working is that it acidifies endosomes and potentially alters the internal environment and blocks the releasing of virus from the host cell. I would like to see specific data that looks really carefully at this mechanism of action. I think it's potentially important as long as it's being tested under controlled clinical trials and not being used anecdotally or widely. I think there's a potential for significant harm if it's not really tested carefully. Certainly, there's at least one example with chikungunya virus where it worked very well in vitro, or in fact, I think in primates or in small animal models. But then in humans, it really caused a paradoxical effect with enhanced replication and decreased immune response against the virus. So there's no compound that can't cause problems, and we need to look at these really carefully. So another area that's been looked at is can we block entry of the virus by blocking cleavage of the spike protein? So there are cleavages, multiple ones that can occur. They can occur on the way in using enzymes such as TMPRSS2 protease, on the way out using furin. This is certainly true for multiple different coronaviruses, and that data will likely be emerging very quickly in terms of what happens when you inhibit those enzymes. But one that has been looked at and has been published recently in Cell was the use of a compound that's been approved for use in pancreatitis, I think in Japan, called camostat mesylate, which is an inhibitor of the TMPRSS2 enzyme, and potentially is capable of blocking virus entry by blocking that activation of the spike glycoprotein on the way in. There are other calpains can also sometimes mediate this, cathepsin. So other inhibitors of endosomal proteases may also potentially be effective at preventing virus entry and replication from occurring. Another place, of course, now what I mentioned is that coronaviruses also synthesize or translate from the genome a large replicase polyprotein with 16 protein domains, and it's like a string of pearls on a chain. One of those pearls on there, or two of those pearls, have protease activity, and those two enzymes are responsible for processing the viral polyprotein into its individual proteins and mature proteins for replication functions. And I'm showing here a schematic of the coronavirus genome, showing what was called ORF1AB, the non-structural proteins 1 through 16, all sort of synthesized as one large or two large polyproteins. And then two proteases, a little blue box called papain-like protease and a little yellow box called 3C-like protease. And then the big blue and yellow boxes show the regions of the replicase polyprotein that are processed by those proteases. So we had data back in 1995, some of the very first studies from my lab were working with protease inhibitors, not for therapeutic principles with mouse hepatitis virus, but to show that they were required for virus replication. And you can profoundly knock down virus replication with protease inhibitors because this process of protein processing is required all the time for the virus. So theoretically, those are really good ideas. And there are some places that have been looked at. So lopinavir ritonavir is a protease inhibitor. I think the data on that is very soft. I think preliminary data data that's been published by Tim Sheehan and a comparison with remdesivir suggested a limited activity to no activity of lopinavir ritonavir in mouse models of SARS-1 or MERS. And so I think that the information for this is not strong. I think this should be looked at carefully, but I think it should, in detail, the data should be looked at whether this is really an active drug at all against SARS-2. There are other protease inhibitors, one that's been used in cats. It's used in cats widely, and it's been able to treat actually a chronic disease in cats, but no known activity yet against this virus. And I'm certainly not being comprehensive here. I want to get on and spend the last 10 minutes or so talking about what we're working at, which is really the issues of coronavirus replication and viral RNA synthesis with the replicase proteins so that it can assemble and modify host membranes into replication factories. And of course, they have an RNA-dependent RNA polymerase. They encode their own polymerase as one of these non-structural proteins that's highly conserved across coronaviruses. RNA polymerases are also conserved structurally across really all RNA viruses, and they're similar to other polymerases in other species. And so certainly, what are the targets? Well, the targets for polymerase inhibition, of course, are nucleoside and base analogs that have been efficacious antivirals against multiple RNA and DNA virus polymerases. And there's some examples here for different drugs. What are these? Well, these are essentially decoys. They look like normal nucleosides that would be incorporated into RNA. And then, however, they have modifications that don't allow they either cause mutations to occur in the genome, or they terminate the RNA chain as it's growing and block normal activity of the virus. So we wanted to look at these, but what was the challenge with coronaviruses? Well, as I mentioned, the challenge is that when we looked at these in the lab over many different years, we'd look at ribavirin because everybody wanted to use ribavirin for everything, and we'd look at other chemical nucleoside analogs that wouldn't be used in humans, like for this purpose, like 5-fluorouracil. And what do we see? So here's what we see. The problem is coronaviruses are resistant to many of these. Basically, if you look at the black lines across the top, these are essentially telling you that there's no decrease in virus titer no matter how much of these drugs you give. These viruses laugh at these compounds. The theoretical curves for inhibition would be the sloping line, showing what we'd like to see. So why is this the case? Well, in fact, we know why it's the case. And the reason is that coronaviruses are the only encoded proofreading exonuclease. This was a discovery in my laboratory by Lance Eckerle back in 2007, and we've studied these enzymes in detail. So these coronaviruses, compared to other RNA viruses, can do proofreading, and they can do error checking, during virus replication. So they can remove misincorporated nucleotides, and they can confer very high replication fidelity, probably 20 times higher than other RNA viruses. So what we've done is when we knock out that enzyme by putting mutations in it genetically and rescue these viruses, we can get a virus back that makes more mistakes. It has a mutator phenotype. And when we then go back and test these same compounds with the virus lacking the exonuclease, what we find is it's now susceptible. So in fact, it is possible for these kinds of drugs to inhibit coronaviruses. You just got to get rid of the exonuclease, or you got to bypass it. So we asked the question: is it possible to have the nucleoside analogs can inhibit coronaviruses in the setting of a proofreading exonuclease? Well, that was a question, and so we started a collaboration. We started multiple ones. We have two major ones that are ongoing. One is with Gilead Sciences, and the other is with the Emory Institute for Drug Development at Emory University to actually test this. And these were based on our really interacting with them and identifying them and having them willing to give us compounds to test. So this is just showing the one with remdesivir, which is the compound which is currently in trials. Really starting back in 2014, where we demonstrated some of its anti-coronavirus activity. And then really up to February of now, when the phase 3 trials began in China. So I'm showing the two compounds here, remdesivir and beta-D-N4-hydroxycytidine, or NHC, and showing that these have what I would call that ideal curve, right? So you can see that you get a decrease. VTR means virus titer reduction, and the numbers -0 through -6 indicate log reductions. So -6 would be a millionfold reduction. And you can see with increasing drug concentration, you see a decrease in virus titer that's log order reductions, up to millionfold reductions. And these have very, very low effective concentration 50s, sub-nanomolar, right? So these are sub-micromolar, low nanomolar concentrations, very effective inhibitors, which we were excited about. And I'll tell you, we still don't know how they bypass the proofreading. And that's an exciting biological question that our laboratory is investigating. How do these particular compounds bypass, block, get by, evade, overcome, decoy? How do they work? Because the discovery for us is just as important from a biological perspective, because it gives us clues about how we could further inhibit other coronaviruses if we understand how these compounds work. So the next step after showing they worked against this, and now we're just going to talk about remdesivir, is to test them against other coronaviruses. And this was done by Tim Sheehan and Amy Sims in the Barrick lab at UNC, where they basically tested this against multiple coronaviruses. So NL63 is a normal human coronavirus. HKU5 is a MERS-like bat virus. We tested them against MERS and SARS as well, against a SARS-like bat coronaviruses. And then circled in red are these what we call pre-pandemic bat viruses. And you can see that basically the curves are all, the slopes are all the same: increasing concentration, log order reduction, and virus replication. So this gave us enthusiasm for potentially working with these viruses. So the ones filled in yellow are the ones that have been tested. We believe it would work against all of them, actually, and based on what we know. And there's evidence in the cell reports out of China that a single-figure preliminary study suggesting that remdesivir is working against SARS-CoV-2 as well. And we have active studies ongoing looking at the details of that, and really much more detailed in vitro and animal studies to test and confirm that. Of course, we're very hopeful that this will be useful. Humans aren't mice. SARS-CoV-2 is not SARS or MERS. And the disease that we're experiencing has a wide range of severity. So at this point, I don't have any data, and then no data available to me. But everything about it from a lab perspective says that it has the potential to work. Whether it works in actual humans remains to be seen. And I can only be hopeful and optimistic about that rather than having any data. So how does remdesivir work? Well, it acts as a chain terminator. Basically, it incorporates into the growing RNA chain, and then it basically acts like a stop sign. It essentially blocks further elongation. So it's one molecule goes in, and that RNA chain stops. So it's very active. That kind of absolute selection would suggest that there might be issues associated with resistance also. And we also wanted to know what happens with proofreading. Well, what turns out that actually, if you get rid of proofreading, it's actually more sensitive to the drug. So it actually is working. It is blocking it. It's not blocking it completely, but this to us was exciting because the drug works very well. But this result suggests that if we could do something to block the exonuclease, that in fact we might be able to really increase the potency of these drugs in some kind of combination form. And that really is an exciting prospect for us to study. So I'm not going to show you all the animal data because I don't have time, but I wanted to say that the studies done again in the Barrick lab and published in 2017. Apologize for skipping the slides there. Decreases the virus replication and virus disease in a lethal SARS mouse model. It's an IV drug. When it's administered before or very early during infection, it decreases virus titer, decreases weight loss, decreases lung pathology, and improves lung function, all of the above. When it's given later in infection in mice, so that's really after 2 to 3 days because the animal disease in SARS is very accelerated. It decreases virus titer very effectively when given at any time, but it doesn't decrease lung disease or mortality. This is what I was mentioning earlier, as we see with other viruses, where it's really immunopathology that's driving the late disease. And so from the very early, one of our goals, of course, as I said, to extend the therapeutic window. And the question is are there host immunomodulators that could be used, I would argue, in combination with direct-acting antivirals that would allow you to use these drugs for longer periods of time or in sicker patients and hope to have a better outcome. And I think that that's something we're very interested in. And obviously, we would have tested all of that before the pandemic if the pandemic had waited. So finally, just a couple other points. Does it select for resistance, and what are the consequences? So I say there's really 2 pieces of good news. One is it was really hard for us to get resistance. We had to coddle the virus, give it all the niceness we could, low doses of drug, take the drug away, put the drug back, give it a day off, all kinds of things before we could get any resistance. And we were able to get some. The other good news is that resistance was limited. It was like less than 6-fold. So it was a really limited resistance. As you can see from our passage 23 virus, it was a little less sensitive. And we identified 2 mutations that we identified that were responsible in the viral polymerase, which is where we expected to see them. So the question was, these viruses looked like they grew pretty well. So the question was, how good were they and how fit were they? How much could they compete with the wild type virus? And what we show here in brief summary is 2 things. On the left panel is basically showing that we took the resistant virus and we took the parent virus and we put them together in host cells. We took both of them and we just let them compete. And then we took the competed cells and we passaged the virus from cell to cell culture. But what we discovered, and you can see here, is even when we give 9 times as much of the resistant virus mixed with the wild type virus over multiple passages, the wild type always won. So this resistant virus was actually weaker and less fit. And in animals on the right, the blue and red curve show what happens when high dose is given of either wild type virus or the mutant virus. And the blue curve just shows that the animals get less sick, they lose less weight, and they recover faster. So this virus is also attenuated. So if resistance arises, it's not competitive, and it's likely to not survive in the absence of the drug. So just to summarize just a couple things. One with remdesivir is it really potently inhibits multiple divergent coronaviruses very well. It has a high barrier to resistance and is detrimental to the virus. It's really useful for prophylaxis in a mouse model and for early treatment. It's likely less effective if it's going to be used late. And so that has implications for how the trials are being conducted. And I hope and trust they're being conducted in a way that you can actually assess its impact on earlier disease as well as very late disease where people are really having end-stage ARDS. Finally, the green tabs on the right are basically showing you that that this other compound, EIDD 1931, 2801, or NHC, also meets all of these criteria in our hands, and that the animal studies are in bioRxiv from Tim Sheehan and UNC. But really, all of our in vitro and in vivo studies have demonstrated this drug's working very well. Its mechanism is different. It's an RNA mutagen, meaning it incorporates and causes mutations and causes the virus essentially to mutagenize itself to death. That's a different mechanism. And we have evidence to suggest that these viruses work in very different ways, which makes you think, of course, of the kinds of things we've seen with HIV, which is treatment with multiple different combinations of either nucleoside analog inhibitors, protease inhibitors as combinations. So finally, then come back into this issue of the use of direct-acting antivirals, which we've studied and will continue to study. We're very interested in this. Chloroquine, Pinavir, ritonavir, favipiravir, all of these compounds, as well as others that might arise. And then to begin to think of these in combinations as monoclonal antibodies potentially become online very quickly to begin to test specific monoclonal antibodies and host-directed therapies. And so with that, I want to thank my team, and I want to thank all the people who are actively treating these patients here in Nashville and around the world. We're all in this together. The only event that I'm aware of in human history that's affected the entire population of the planet at one time. It's serious. We need to be really dedicated to continuing to find ways to try to intervene in this. And we are definitely committed to that process. And so I'm happy to take any questions that you might have.
Thank you, Mark.
Sure.
Oh. Thank you, Mark, for that informative presentation. We will now start the Q&A portion of the webinar. And if you have any questions you'd like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen. Okay, let's get started. We have so many great questions coming in already, Mark.
Do you mind if I just sort of pick a couple? Is that okay with you if I sort of look at these?
Sure. If you want to take the lead on that, I was just going to ask of you, but go for it. Absolutely.
Yeah, if I may need help. There's a lot of good questions here. A couple I think that came in really quickly are why are children not getting as sick with this? I'm an infectious disease person. I've watched this forever. I think about this related to other viruses. It's always best to use models. The answer to almost everything about this virus is we don't know because we've never encountered anything like this before, I'm aware of in human history, okay, where the entire planet is immune susceptible. And that's a scary thing because it says a lot about us not really understanding it. But there's some models we can think about with children. And perhaps they're better as evolutionary models than they are as virological models. There's many ways to propose why kids aren't as frequently infected or as ill. Those could be that the receptor specificity is different, that there's developmental changes in the receptors and how they occur on children, that their immune response is actually more naive or less developed, and so they actually may have virological replication, but they don't develop the immune-mediated diseases. That's possible. From an evolutionary perspective, it's almost a tautology to say that, in fact, they don't get sick because they don't get sick. In other words, that over time, that human beings have been exposed to so many serious viruses over time that there's been a development of essentially for survival of the species, you had to have children capable of surviving to adulthood. And so the selection against a virus causing this kind of lethality in children. I say that because there's models, if you take, for example, measles virus, and you go to an immune-naive population like a separate island somewhere, and these have been done. We've seen this with other diseases like smallpox and measles, where they've entered into a particular place like the Faroe Islands. And what you saw was really normal kind of childhood disease in children, but you saw profound or lethal severe disease in adults who had never seen the virus as children. And so, like I said, that's not an explanation, but that's sort of an evolutionary model for how our long-term battle with viruses over the course of our evolution may have impacted how what's going to happen to children when they encounter these kind of new organisms and pathogens. Which brings me to, yeah, go ahead. Mm-hmm.
Well, go ahead. I'm sorry. I was just going to say that one of the common questions I keep seeing in these wonderful webinars today is whether or not the blood type link is linked to coronavirus infection. And I see 3 people have asked that question here for you as well. I think people are trying to find, is there a formula almost, right?
My bet is that this virus has our number, and that there's I think with the diversity of what we're seeing, I would be profoundly surprised if there was a single genetic determinant. There's no sex, racial linkage. Now the numbers are large enough that I believe will explain this. I think this is about the virus. Okay, and about the potential about receptor expression and the potential variabilities that might occur in our immune response. Certainly with some other viruses like norovirus and everything, for example, blood type specificity is important. Some people don't get ill at all. So I think that that's a possibility, and that's an interesting idea to explore, whether there are, HLA types or blood types that might be relevant. That's an exciting research question. Right now, there's no data to support that one way or another. And another question I saw here was why the human being has not encountered this type of virus before in evolutionary history, if the interaction with wild game was even greater than currently. I would argue it has. I would argue we have encountered these all the time. And the question is does it have all the capabilities of maintaining itself in our population and transmitting? So what's changed? What's different? Well, I think in the past, if these encountered these might have been in a small village, in a small town. Before coronavirus were known, they might have stayed local. They might have been in the background of other serious illnesses like that were prominent and maybe now more vaccine control, like pneumococcus, influenza, diphtheria, you name it, tuberculosis, other things. And so the idea of a single death from a pneumonia would never have been identified, or even a cluster, or even a small village. But those didn't have, what didn't they have? Well, they didn't have the capability to travel. They didn't have the capability for a large number of people clustered in major urban centers. They didn't have time to develop other things. The other thing is, I think this is maybe a little bit about the virus. It's just about the virus because there's not a lot of, it does not look like there's a lot of selective pressure on this virus for evolutionary or adaptive change. It looks pretty much ready to go. It kind of came out with its Swiss Army knife of tools and it's ready to go. And I think that's different. So once the encounter occurred and the opportunity occurred, that it was human demographics, human travel, whether that was in China or elsewhere, that allowed this virus to disseminate, as well as its sort of stealth modes. I had been arguing from the very beginning that this virus was likely having asymptomatic or pre-symptomatic transmission. So all of those things may be why this is sort of the perfect storm that we have been worrying about.
Dr. Denison, I want to just pause and I want to thank the audience because we have over 40 questions coming in, and I know that we're not going to have time to answer all of them. And I just want to remind them that any questions not answered today will be answered via email. And I noticed quite a few questions asking about the AG detection, about the sample of choice, and also about the procedures in the testing. Maybe you want to touch up on that, maybe?
I could. I'm not the expert on that. I think the okay. Standard tests that have been coming from CDC here certainly have been based on the nucleocapsid gene. So that's this particular region of the genome that's been identified. I think that it has specificity for sure, and I think that in our hands are others that the kind of assay has sensitivity associated with it. I'm not able. I don't have the data on denominators. I also don't have the data on controls, positive or negatives. So the issue, I think, in a high prevalence time, when you have a setting where there's a high probability of the infection being there, that tests like this can perform very well, right? Because you're unlikely to have false positives or false negatives. Because if you have a consistent syndrome, or you have a population that's highly exposed, then that's really not so much of a concern as if you're looking for something really rare in a population and your test may fail. So I don't have a lot of data on that. I probably can't go any further with that, but I have reasonable confidence that these tests, I have good confidence that these tests are performing as advertised. The rest of it's about the virus again. So there are now data coming out that PCR positivity may drop off pretty rapidly in a real infection. And so it really depends as much on when you get it and how fast you get it and if you repeat it in certain circumstances as it does with the ability of the test. So I actually think the test is good. I think it's the virus that's showing the variability.
Mm-hmm. And is there any indication on how often the virus mutates? Is it similar to the flu mutation?
So there's not a lot of data for mutation. I think this is really, really important. There's not a lot of data for mutation yet. I think the pressure for when will the pressure for mutation arise? In SARS, that appeared to happen pretty fast because it looked like it was ping-ponging back and forth between humans and civets, okay, and other animal vectors until it finally adapted to humans. And then there was adaptation to in humans with other changes that had to do with its binding to the ACE2. This virus has, and I am not up to date on the thousands of now specimens available, so that please take this as a caveat, but it doesn't look like it's doing that yet. When will that pressure, if it's not doing that yet, when will that pressure arise? That pressure may not arise until you have a substantial amount of either people have been vaccinated or that you have a substantial number of people who've had natural disease and now have some or existing immunity. then there will be selective pressure on the virus for change. Until then, it doesn't look like it's under a lot of selective pressure, which is scary, I think, because it means that it's capable of moving through the population without having to really adapt as it goes. The only advantage of that is that means that vaccines that might be developed now might be in a position to go against all of the potential viruses that are out there. But people are talking about circulating strains. I think that's not as accurate a phrase as I'd call them polymorphisms, viral polymorphisms, because those don't appear to be really impacting this disease of the virus so far, or a probability that a vaccine that's developed, a single type of vaccine that's developed, would be useful. I think about it's more like measles, where there might be some variations that occur, but the vaccine still works, at least initially.
One interesting question coming in. Estrogen helps with immunology protection. Could this be why there is a difference between men and women with their severe infection rate?
I haven't seen the numbers. I was unaware that there may be some isolated things in terms of severity of disease, but some of these can be masked and be wrong. For example, with MERS, there definitely was a sex distribution with more men, but that also had to do with the fact that men were more likely to be around camels, right? So there was something, there were some things that looked like they were sex differences, but really weren't. They were more about exposure differences. So my hypothesis would be that we're actually not going to see sex differences in severe disease. We might find out there's more to do with pre-existing conditions, other underlying conditions. And this is the challenge of really getting good enough epidemiology. I wanted to answer, if I could, a science question again. For at least one person asked me a question about remdesivir gets incorporated during replication. It adds 3 bases, then replication terminates. That would explain how it gets around the exonuclease. Can you comment on this? So thank you, viral geeks. I like those questions. Right in my ballpark. So the point here is, what they said is, my model showed that it incorporates in the RNA chain and it terminates. That would be called an obligate chain terminator, meaning it has to stop right away. This person is absolutely correct that this probably incorporates the nucleoside, and then it adds 3 more bases, and then it terminates. So it's called a non-obligate chain terminator, which sounds like a oxymoron. That is one possibility for how it gets by the exonuclease, because that if the exonuclease is always checking for mistakes, if it's checking right as it gets incorporated, if the thing has already moved on 3 bases, that it might not know it's there. So coronaviruses might not have that capacity. And that's an excellent question. And I think we'll go back and look at that in more detail. There are biochemical models coming online. But this is really important, because if we're going to develop multiple compounds that are effective against all coronaviruses, this one and going forward, that we need to have that kind of understanding of the details of the biology.
Absolutely. And another question, could you elaborate, if possible, is on the COVID being airborne in high temperatures based countries. For example, this particular audience member is in the Caribbean. Yeah.
So this comes to this issue of seasonality. I like to say viruses, especially viruses, aren't seasonal; they're migratory. They appear seasonal to us because we're hemisphere centric, right? And so flu, if you think about it, we're always picking the flu vaccine based on, for the North America, we're picking the flu vaccine by what's going on in Australia or China or elsewhere in the world, right? In the other seasons. And that's true of seasonal coronaviruses. It's true of respiratory syncytial virus. These are viruses that seem to migrate. But in the tropics, they tend to not be seasonal. They tend to be low-level endemic year-round, not necessarily causing the peaks of epidemic height, but kind of consistently present. The concept that this virus will become a seasonal virus maybe happen in a year. It may take 1,000 years before it becomes a seasonal virus, as long as we have a significantly immune naive population. I would hope that's not the case, but it's already in the tropics. It's already in the southern hemisphere. And so viruses like humidity, and certainly in the lab, they like humidity and temperature. They like 37 degrees centigrade, and they like lots of humidity. They grow very well, and they survive a long time in those kind of environments. When you get to certain temperatures, they become more labile and damaged more easily. But I think it's much more complex than that. It has to do with what kinds of materials they're in, what their stability is on environmental surfaces, what's the difference between saliva and nasal secretions, and blood and sputum and different things like that. Can they be airborne? I think the data coming out suggests that they can survive as airborne small particles for multiple hours. I don't think they'll ever be considered like measles or chickenpox, where if you're in a room 2 hours after someone's been in there with those diseases, you can still get it. They're not like that, and they're not like TB. But can there be transmission by airborne routes? At this point, I think the data suggests yes. I have one. What are you testing right now? We're testing the compounds that we've already tested, NHC and remdesivir. We are certainly looking at other combinations. We're very interested in protease inhibitors of multiple kinds. We're really interested in novel mechanisms that might affect virus entry or virus exit. There are multiple other proteins that the virus makes. They make a polymerase, an exonuclease, a helicase, a TPase. They make multiple processivity factors and RNA binding proteins. We're interested in screening, but we're really interested in targeting those kind of enzymes as well. Certainly, we're interested in targeting the exonuclease. So, if you ask me what's one of the most important things to do this, I would say maintain programs and build up manpower of people and young investigators who can be supported to do this kind of work. We have a small lab. We have 5 or 6 people who can do this work at BSL-3. It takes a long time to train people for this kind of work to do it safely, effectively. So this is a limiting resource. We are a genetic bottleneck to management and treatment to testing against the virus, us and a few other people. So I think this points out the need to continue to prepare like we've been trying to prepare for the viruses that come along right now.
Thank you very much. We have time for a couple more questions. How can we choose specific molecular tests from the ones available from different countries?
I think this is another diagnostic question about what kind of test we want to use. I think the idea is we want to develop multiple ones. For example, I had a question from someone today who had been infected, and they want to know, so what's my probability now that I and my family have been infected, and I think my kids are infected, my wife is infected, and we're all recovered. Can we be with people who are ill? Can we be caregivers for those people, for their children, now that multiple families are involved? Can we be around them, and are we safe? Or are we going to get reinfected, and maybe we won't get ill, but we'll be transmitting virus? And I think
that's an important question.
The point is that we need to have an initial test that rapidly can be used for both diagnosis and for surveillance in epidemiology. And second, we need to have tests that can allow us to understand how are people protected. So serological tests to say, does their immune response, their secretory IgA, their circulating IgM and IgG, are those neutralizing? Do they kill the virus? And are those people actually protected from reinfection? Likely protected from reinfection. What's that going to look like? Coronaviruses are, the bad news for coronaviruses is that immunity does not appear to be highly durable. That is, I can get a respiratory coronavirus in year 1, another one in year 2, but I can get A again in year 3. It's usually milder. It's not as severe, and I think it probably has some protective effect, but it's not 100%. So it's important for a disease like this because you don't want to be in a position of not being ill, but having the virus and shedding it to someone else and maintaining the epidemic.
Thank you so much, Dr. Denison. And again, I want to remind everyone that any questions not answered today will be answered via email. Did you want to provide our audience members with any closing remarks?
I think we're all in this. So I'm afraid I'm possibly my age and possibly working with these viruses for so long. I think we're all in this together, and that we have to have these multiple modalities. We have to not be just thinking about now, but the future. I think this virus is with us for some time to come, and its implications are profound. I'm interested in talking to people about this and mechanisms that we could use to have combinations of therapy for, if you want to lobby the NIH or others for the need to get manpower both in the treatment but also in the research components of this. I would be happy to chat with you about it. Thank you, and thank you again for your time today and for your important research.
And before we go, I'd like to thank the audience for joining us, and again, for your interesting questions. Questions we didn't have time today will be submitted, you can submit on demand, and will be addressed by the speaker via your contact information you provided at the time of registration. And a reminder, this webcast can be viewed on demand through the end of the year 2020. LabRoots will alert you via email when it's available for replay. We encourage you to share your email with your colleagues who have missed today's live event. Until next time, bye-bye, and please be safe and healthy. Take care, everyone.
Okay.